Thank you. First of all, I'd like to thank the organizers. This is really a great honor to be telling you about a bit of our research. I have to say that all the presentations have been very, very exciting and thought-provoking, and it's been really nice to hear and discuss about the results related to the early life period and the epigenetics related to that. So yes, so we kind of tell in the title already that we did not find any large findings with respect to the methylation and maternal anxiety during pregnancy, but hopefully I have some good news also to tell to you. It's really pleasure to continue from the perfect lecture that we heard earlier on the developmental origins of health and disease. I had an honor to work with late Professor Barker when I was doing my PhD. He did a very, very close collaboration with one of the cohorts that we have and the study groups that I'm also part of back in Helsinki, called the Helsinki Birth Cohort Study, and have many, many nice memories of working with him with respect to obviously to the early life anthropometrics and various outcomes or various child and adult characteristics afterwards. So this is the framework that we're working also with, so the DOHAD framework, and in short, I think this kind of tells very, very nicely what it is about. So it's about adverse influences during sensitive periods of development that change or program the structure and function of the cells and organs, and consequently the function of the organism. And these changes persist throughout the lifespan, or at least for a very long time. And as we can see here, there are different exposures that would lead to variation in the birth size, for example, and many different kinds of outcomes. So this is the general framework, and we've been mostly interested in maternal mental well-being during pregnancy, and this is a nice review back in a few years ago by Kieran O'Donnell and Michael Meany, and this shows the situation with respect to the most common disorders during the pregnancy, so pregnancy-related depression and anxiety and stress also. And it basically tells us that we have most literature on the associations between depression, maternal depression during pregnancy, and neurodevelopmental outcomes of the children, and also quite a lot of research showing that also stress during pregnancy links with child neurodevelopmental outcomes, and a little bit less on anxiety during pregnancy and the child neurodevelopmental outcomes. And what it also tells nicely that we have nice research showing that, as we heard in the previous talk, that HPA axis system glucocorticoids seem to mediate these effects, and also placenta has an important role, as we heard, with respect to element beta HSD2, and back from the David Barker studies, we know that birth weight is in the kind of the one link in the chain also there. And also they very nicely tell us that there are moderating factors such as gender and genotype also in the chain there. We've had a nice opportunity to study these early life maternal mental health issues and their effects on the kids in the PREDO cohort. So that is the cohort of about 4,500 mother-child duets that we've been working with since early 2000. And we've followed the mothers from the very early pregnancy, so from the week 12 more or less throughout the pregnancy, and the poor mothers were asked to fill in questionnaires every 2 weeks. So we have a average of 14 sets of questionnaires filled in by the mothers during the pregnancy. So we can pretty much focus on the different parts of pregnancy and say whether there are differences in the timing of the exposure. And here we can see, for example, that the mothers whose children had behavioral problems in childhood had higher scores in the depression scale throughout, more or less throughout the pregnancy, which is the higher, the lines here. And it doesn't really kind of show big effects of timing. There is somehow there's a small trend towards the later, the third trimester of the pregnancy is showing a bit more stronger effects, but it's not actually statistically significant. Whereas the mothers whose children were not considered as having behavioral problems, they scored lower throughout the pregnancy in terms of the depression. We have other types of evidence showing more or less same kind of results. Also, for example, here we show that maternal depressive symptoms throughout pregnancy also associate with diagnosis of mental and behavioral disorders in the children. So we have an opportunity to also use the wonderful registry system that we have in Finland. So we have basically all the diagnoses from the hospital and actually from the outpatient units also nowadays of the 3,365 children. And with these data, we also, or in this study, we also looked at the maternal anxiety symptoms during pregnancy and also maternal stress during pregnancy. And the results look pretty much the same as with the depression. So even though there are less studies on maternal anxiety during pregnancy, it seems to affect more or less the same way the children as the depression or stressful life events or stress in general. So this is the kind of the line one here that we've been discussing about now. So the direct effects of the maternal exposures. We've been interested in and focusing on other types of exposures during pregnancy also, such as the environmental chemicals, which I'm personally very interested in at the very moment. We have really nice results there, and also folate and CRP and thyroid hormones and some other stuff also. But what I'm also very, very much interested is the mediating level of epigenetics in between the exposures in the mother and the child outcomes, and also the moderating role of the genome for this association. So we're looking whether we find genes that could kind of moderate the mediation effect that we are seeing there in the data. So in the cohort, even though we have some 4,000 with questionnaire data, we don't really have quite that many, unfortunately, with the genotype data and with the methylation data. But we do have pretty nice sample with those kind of data also. So we have cord blood methylation data with 450K and with EPIC from about 1,000 children, and we've been very much doing collaboration with Elisabeth Binder from the Max Planck. And we do have follow-ups from 2 weeks up to 10 years, and actually we are following the children up and the families up at the very moment also. And this is one of the wonderful results that we've had linking epigenetics between the prenatal exposures and the postnatal child mental health. So my PhD student, Ana Suarez, nicely showed in her work that, first of all, as we already saw, the maternal depression also in the smaller sample with the methylation data. So this is about 800 mothers and children. In this sample also, the maternal depression during pregnancy was linked with with child internalizing problems at the mean age of 3.7 years, so about 4 years. And also that maternal depression during pregnancy was linked with child epigenetic gestational age at birth. So this is the same type of, or very similar type of methylation-based indicator as the Horvath epigenetic clock, but Horvath clock works best in adulthood and in late adulthood, whereas the epigenetic clock also is much more fine-tuned towards the gestational age. So Horvath clock is a little bit more robust in a way that it kind of works in a long time periods, but when it comes to a very short period in terms of the human lifespan, such as the period of pregnancy and the gestational age, obviously you need a bit different kind of indicator. So we've shown that the Horvath clock actually doesn't work as the indicator of gestational age very well, whereas this indicator has been fine-tuned for the gestational age. But what's the kind of the take-home message for this analysis is that the gestational age epigenetic clock, if you will, mediates the association between the maternal depression and the child internalizing problems. So it seems like there is an indirect path going from the maternal mental health affecting the cord blood methylation in the child, and then continuing to the child mental health at 4 years, which I find really very, very exciting and inspiring. We have another kind of example showing that methylation in cord blood is really important. Provençal and Elizabeth Binder a few years back published a paper in PNAS with an idea of whether we could develop a methylation-based indicator or a score reflecting exposure to glucocorticoids. And I think I find this also a very, very nice idea, and we were happy to participate in this study. So first they developed a set of CpGs in methylation based on dexamethasone-treated hippocampal progenitor cells, and then also blood cells from the human cohort, MPIP cohort. So this was a set of 496 CpGs, and then they used this lasso method to select 24 CpGs out of these 500 to form a score. And then we tested in the pre-dough whether this score links with the maternal depression during pregnancy and anxiety during pregnancy. And we were very happy to find that it indeed does. So it's not only a score for current exposure to depression or linking with the current glucocorticoid exposure, but it also seems to work with maternal depression and maternal anxiety during pregnancy and reflect those kind of exposures. So there is a hypomethylation with the higher scores in the depression and anxiety questionnaires. And then we checked whether this score has any relevance with respect to child outcomes later in life. And again, my PhD student work, she looked at the child diagnosis, mental health diagnosis in later life, and this time I think we had data until 8.5 years, so much more diagnosis than earlier. So in this case, we have 99 child with mental or behavioral disorders in the sample, and we it's still rather small sample, but still, I mean, we didn't find associations directly with the disorder itself, but we found a nice link with the length of treatment for the mental health disorders. So the ones who showed lower scores in the glucocorticoid exposure score that was, as we remember, that was linked with higher exposure to maternal depression during pregnancy. They tended to spend more time in hospitals or in treatment because of their condition, because of their problems, which I also find really nice. So this is kind of a predictive validity study for the glucocorticoid exposure score based on methylation. I'm really looking forward for other results, also other studies related to this score. I think this is really promising, and it will show most probably very, very interesting results in the future also. So, and then getting to the topic itself. So what we kind of see in the literature now is that we do have rather nice results on the maternal depression during pregnancy, and in its links with child outcomes, and also some evidence that mental health, this link could be mediated through epigenetics. But with respect to maternal anxiety, we don't really know that much, but we do have some kind of early stage insights that it might work the same way with the anxiety also. So in this situation, we decided some years ago in the PACE consortium that we should start a working group on maternal stress during pregnancy, and focus on different types of kind of stresses defined in a very, very kind of a wide way. So we chose to focus on maternal depression and maternal anxiety and maternal stressful life events within the PACE consortium. And I'm leading the working group together with Kelly Brunst from the University of Cincinnati. And we have a really nice collaboration that has continued for many years now, and within the PACE, and also expanded a little bit too. So PACE is the consortium working or focusing on very early life, pregnancy-related and early life-related epigenetical studies. I think it's most well known on the pregnancy-related exposures and the cord blood methylation. So most of the cohorts that are part of the PACE consortium do have data on those exposures and methylation in the cord blood. We also have some studies on the childhood methylation, and we have another cohort called the GLACU cohort with childhood methylation data. But unfortunately, in this presentation, I don't have time to go to these results, but would be nice to show some of those too. But yeah, so this is the first of our series of 3 papers that we're about to publish with the PACE consortium that I'm presenting you in this presentation. And obviously anxiety is very interesting exposure during pregnancy, also in a sense that it is really common. It's actually more common than depression during pregnancy and depression in general. So about 15% of the mothers during pregnancy at some point could be diagnosed with anxiety disorder. So this is pretty common disorder. It is also linked with preterm birth and small for gestational age status in the child, and also with the high risk for emotional and behavioral problems in the child. And once again, the question arises whether there could be an epigenetic mechanism linking those exposures with the child outcomes. And the first part of to kind of show that obviously would be to show that there is indeed a link between the maternal anxiety during pregnancy and some methylation changes in the cord blood or in the child in general. And obviously there are bunch of mostly candidate gene studies out there showing these effects, but but these are rather small studies at the moment, and they are focusing on the kind of the obvious candidates, most often the HPA axis related genes. And I find that this is quite the same situation that we were like 15 years ago with the GWAS studies, that with the core genomic studies, they started with the candidate gene findings, and all the researchers had their favorite genes and favorite systems to look at, and I was part of those studies, and it's a little bit kind of a even funny nowadays to look at the studies that we published with very, very small samples and very, very focused genotyping. First of the two first studies that I've written were gene environment interaction studies with 150 people focusing on dopamine receptor D4 back in, I think it was early 2000 or so, and we were able with the 150 people show some gene environment interactions, which nowadays would not be possible to publish and would be kind of a bit funny and ridiculous to publish. But anyways, back at that time, it was really nice sample, and it was very, very cool to work with those and show those results. And I've kind of find that we are in the very early stages of the genome-wide epigenomic studies at the moment. So we also have a few quite small EWASs, epigenome-wide association studies out there on the anxiety, and they do show something, but still with a sample size of well, sometimes tens and sometimes hundreds. It seems rather unlikely that the findings that we see in these studies are very, very strong ones in the future, or very kind of meaningful ones in the future. At least that's what happened with the GWAS studies, that there was this huge hype with the first GWASs. They didn't show almost anything, but when we now look at the peaks that we saw in the first, say, schizophrenia GWASs with rather small sample sizes, it seems like the same peaks have grown to reach the level of statistical significance and after the correction for multiple testing. So I do have a hope that some of the trend level findings that we are also finding at the moment will show a stronger effect or significant effect in the future when we have more samples. So this is kind of preparing you for the null findings that we have in this study, and giving kind of a hope that even though we didn't find much in this respect, there are issues with the design of the genome-wide studies that make it very difficult to find something if you don't really have a large sample size, as I'm sure that all of us know. So with the maternal anxiety and methylation that we were focusing on, we had 16 cohorts. This paper was, we were actually a bit surprised and happily surprised that it was published in Molecular Psychiatry last year, even though it's a null finding paper. And we had a really, really nice discussion with the editor and with the reviewers about the different kinds of ways how to analyze the data and reanalyze the data in a way to search for whether there are any findings. And we did everything that we could do to analyze, but came up with nothing. And it was really nice that they still published the study. I mean that quite often the null finding papers end up in a little bit kind of a lower quality journals. But anyways, we have the largest EOS study today concerning prenatal depression, concerning prenatal stress, prenatal anxiety, you name it. This is the largest at the moment. So we have a sample size of 7,243 in the study, and 16 cohorts, 12 independent longitudinal studies all over the world. Most of the cohorts come from the European ancestry populations, such as the ALSPAC and BIB, and Eden from France, Generation R from the Netherlands, Goya from Denmark, Inma Spain, and obviously MOBA from Norway, and our cohort Prido from Finland, and then we have Prism from US and Project Viva from US, Progress from Mexico, and so forth. And this is a work of my or our former PhD student who is now doing her postdoc in, or actually she was doing her postdoc in the Netherlands during the time of this paper. And this is very much collaboration with Henning Thiemeyer, also from the Erasmus MEMC from the Netherlands. And this is the workflow that we have in the PACE. So sure, perhaps you already know that typical workflow, and that this is very similar to GeoAssist that I've been working with like 15 years now or so. But this is the same kind of workflow with the methylation, genome-wide methylation chain data. So first we propose a project like maternal anxiety or maternal depression. We write an analysis plan for the consortium. Studies decide whether they are in or out in the analysis, and then they run the cohort-specific analysis, provide us with the summary data. We do some quality control on the summary data, and quite often get back to the cohorts and ask for more analysis or some other kind of changes in the analysis. Then we meta-analyze the data from all these 16 different cohorts. We also, it's very typical in PACE. It's not that typical in other consortia, I find, to run the shadow meta-analysis. So we kind of rerun all the meta-analyses by an independent people to make sure that what we find in the meta-analysis really hold, and there are no mistakes in the meta-analysis stage. And we try to replicate the findings in some cohorts, or if we find new cohorts, and we do some functional stuff also. And finally, the article. So in this case, we focused on two types of maternal anxiety during pregnancy. First was, we named it as general anxiety, and as we can see here, it's been measured with different kinds of instruments during the pregnancy. There are 7 or 8 different types of questionnaires on maternal anxiety. Then the other type of exposure or type of anxiety was pregnancy-related anxiety. And once again, we have 4 cohorts there. We have 4 different kinds of measure for pregnancy-specific anxiety. Even though there are many different ways to measure it, but I find that pregnancy-related anxiety, the way that the different questionnaires are measuring it, is pretty consistent. So it is much more homogeneous exposure compared to, for example, to general anxiety, which is a little bit more heterogeneous. I'm just trying to, I'm sorry to bore you about the methodological details, but I think these relate to the discussion on why we didn't find these findings. We also did 2 different kinds of exposures. So the first one to kind of mimic the fact that about 15% of the mothers are diagnosed with anxiety disorders during pregnancy, was that we categorized the continuous questionnaire variables into 2 categories. That one's show the mothers showing high anxiety symptoms and the rest of the mothers, and we used the 15th percentile as a cutoff. And then we used the traditional continuous exposure cutoff as a secondary analysis also. We looked at the Illumina 450K CPGs and combined the EPIC CPGs with those. And then we looked only at the EPIC, those CPGs that are only at the EPIC. And as you can see, there are a little bit more even those only EPIC CPGs than in the 450K chip. Then we had a harmonized cohort-specific normalization and some QC measures. At the same time, some of the cohorts had their specific QC processes also that we couldn't really kind of affect anymore. And then we did some other other QC procedures also to remove bad CpGs, for example. We used robust linear regression, which should be pretty well suited in this case. We used 2 sets of covariates, child sex, maternal SES, maternal age, ancestry, batch covariates and cell counts, and in the other model we also adjusted for maternal smoking during pregnancy. In the sensitivity analysis, we looked at the only 450K cohorts, only EPIC cohorts, and only European ancestry cohorts. So I'm just telling you because I'd like to show you that we really did everything we could think of, and everything that the editor and the reviewer could think of in terms of looking at the data a little bit different ways to find out whether there were some biases, for example, that would mask the true signal there. So this is all that we could think of. Please tell me if there's something else that we should have done. And then we ran the meta-analysis as sample size weighted fixed effects meta-analysis, very typical for GWASs and EUSs, nothing really special about that. And we took account on the heterogeneity also in the analysis. We used the Bonferroni cutoff. This can be debatable. There are different estimations on a little bit higher cutoff for the 450K chip analysis, but as we had also the EPIC chip, we felt that we should be using the traditional Bonferroni correction analysis. We did some DMR analysis also, but I actually don't go into those in this presentation. So this is the first results slide, and as you can see, we are very we are pretty with this is the Manhattan plot, chromosomes on the x-axis and the p-values on the y-axis, and the red line indicates the significance level for Bonferroni corrected result. And as we can see, that the smallest p-values are around 10 to the -6, which is really very far from a significant effect with respect to the general anxiety. And here in the analysis, we have a sample size of 6,686 participants out of 15 different cohorts. And we were lucky to see a very nice QQ plot here, so there's no technical issues with there shouldn't be any technical issues with the data, which is not the case in very many EUS analyses, as there are many technical issues that could affect the results, and sometimes you see really poor QQ plots that unfortunately the editors do not ask to correct for. Okay, so here's the results concerning the top CpGs that we see, and I'm just showing you the kind of the first genes that we have, the smallest hits, even though not significant. But this is the KIAA 0182. Have you ever heard of this gene? I haven't. And p-value 1.8 to 10 to -6. PDE8B, ACT1, and ZC3H7A. The good news is that most of these are the results. This is a forest plot of results based on different cohorts, so by different cohorts. So we do see rather consistent pattern across the different cohorts, which at least tells me that if we had more cohorts, if we had more data, we probably would reach significance at some point. But even with 6,600, that's just not enough at the moment. Then with respect to pregnancy-related anxiety, we see more or less the same pattern here with 2,000 samples from 4 different cohorts. And once again, we have a really nice QQ plot. Manhattan plot shows that the smallest p-values are pretty far from being significant. And when we looked at the annotation to closest genes, this is TRIL, and this is KCNIP4 here. And when we looked at the EPIC only CpG sites and a sample size of 1,221 in 4 cohorts, we have the smallest p-value, the most the site with the largest effect here, closest to the red line there, which is here in the MCF2L. Then the next one would be in CACNA1H and trio. And the reason why I'm showing you these genes is that, well, at least those genes were not among the list of my favorite genes. Not sure if, please tell me if they are among the list in your, and this is once again, this is very, very similar situation as we had back in the old GWAS days. So we got hits here and there, and they were most often not in anyone's list of favorite genes. So they were really, really surprising genes to figure out, telling that probably the biology behind the anxiety also, we just don't know, we just don't understand it good enough. I'm sure that HPA axis will pop up as with the depression GWASs, it will pop up eventually at some point, but it's not definitely the only system that is linked with the anxiety, and probably there are much more important in systems in a way that they are affected earlier than the HPA system. And here we did also some favorite genes, candidate genes, lookups from our EOS results, and here are those. So we just wanted to point out that indeed, if we look at we took all the genes that are within the kind of a reasonable HPA axis pathway, and the smallest p-value was in FKBP5, CPG in FKBP5, and it shows something like 0.01 level of significance, which is really, really far from 10 to the -7 line. So conclusions: no significant associations between maternal anxiety during pregnancy and cord blood methylation, and we have discussed, or kind of already discussed some of the reasons for that: small effects, small sample sizes, heterogeneity in exposures, although we tried to overcome that by looking at 2 different exposures. We may have wrong timing of the exposure, or we should probably look at the chronicity of the exposures. There could be some modifiers such as sex of the exposures, or maybe some other comorbid conditions such as depression, which is very typically comorbid with anxiety, or maybe some of the very kind of dysfunctional coping skills that are or ways that are linked with anxiety, such as drinking alcohol or other substances. Or maybe we should look at the protective factors at the same time. I mean, it's also always a combination, as we know. Subjective stress comes from the primary appraisal of the threatening situation, and the secondary appraisal of whether I can cope with that. Maybe we should include the secondary appraisal factors there also, which are like protective factors. Maybe we are just have heterogeneity in processing the methylation data, although we tried to work on that too. Maybe we have the wrong CPGs. We're only covering 1.5 to 3% with the 450K and EPIC chips. Maybe we have the wrong tissue. Maybe we should have looked at the placenta, for example. Maybe we have a totally wrong type of epigenetics. Maybe we should be looking at the histone modifications, or maybe there just isn't any results to find in the first place. So this is actually the repetition of the earlier slide. And some future prospects with respect to stressful life events. So we're continuing those manuscripts, those papers, and with the stressful life events. We do have a sample of 12 cohorts and 5,500 people, and we have 5 different stress domains that we have harmonized across the cohorts, and we're looking at those, the effects of those on the cord blood methylation, as well as the aggregate stressful life events score. And then we have a little bit larger sample size with 7,500 in the maternal depression analysis, and those papers are out soon. Thank you very much, and thank you for being able to present that, and thank you for the Memory Foundation, and there are the acknowledgements on our team of group, and these are more or less the same people a few years earlier, and I'm actually one of these guys here too, and thank you for your attention.